Hi, my name is Sarah Wolf. I am currently finishing up my PhD in Kim Roswell's lab at Indiana University, and today I'll be sharing my work on the predictive power of the shelterin protein POT1 on performance in a songbird. There is an incredible diversity of life histories in senescence, from the 24-hour life of an adult mayfly to the hundreds to thousands of years old black coral. To understand the evolution of senescence, it's critical that we explore the mechanisms underlying the aging process, as this is the fodder upon which natural selection can act. Now, one established biomarker of senescence is the telomere. Telomeres are conserved sequences of DNA found at the ends of chromosomes that form a protective cap-like structure. These telomeres naturally shorten during cellular replication and therefore with age, a process that often becomes accelerated with exposure to stress. In the last decade or so, telomeres have quickly emerged as strong biomarkers of senescence because across many taxa, short and/or shortening telomeres often predict higher mortality and shorter lifespans. What remains unclear is what mechanisms link telomeres with variation in senescence. Biomedical work suggests that telomeres can orchestrate direct changes in senescence-related gene expression, but telomeres are more often thought to simply correlate with other mechanisms that drive senescence, which may explain why telomeres don't always relate to fitness. A large body of work has already begun to explore the roles mechanisms like metabolism, oxidative damage, and antioxidants play in regulating telomere dynamics. However, one relatively unexplored set of mechanisms that could explain the telomere fitness link are shelterin proteins. These diverse proteins are responsible for maintaining chromosomal stability and facilitating conformational changes at the telomere end, which essentially opens and closes it to replication and repair. For example, shelterin proteins physically interact with the enzyme telomerase to either prevent or facilitate its ability to lengthen and rebuild the telomere end. Interestingly, shelterin proteins can also regulate body condition via metabolism, immune responses, and cellular replication. Shelterin proteins could therefore grant telomeres their biomarker status because of the pleiotropic roles shelterin proteins play on both telomere length and body condition. A rich biomedical literature has shown that dysfunction of shelterin proteins is intimately linked to cancer, the extremes of variation in lifespan. However, whether natural variation in shelterin proteins maps onto fitness and senescence in the wild is unexplored. For this presentation, I will focus on protection of telomeres 1, or POT1, which inhibits telomerase from lengthening the telomere. Like other shelterin proteins, low POT1 is linked to cancer, likely due to out-of-control telomerase activity. However, naturally lower levels of POT1 may contribute to adaptation by positioning the telomere for repair. So I'll explore these ideas by asking whether POT1 predicts performance in tree swallows, a songbird that breeds here in Indiana. Specifically, I'll ask whether POT1 predicts reproductive quality and behavioral resilience to stress in adults, as well as survival to fitness milestones in chicks. For this project, I also used an information theoretic approach in order to ask whether telomeres or POT1 better predicted each of these performance traits. So for the first question, I performed an observational study to assess whether telomere length or POT1 gene expression better predicted reproductive qualities in females, including first egg date, which is a proxy for timing of breeding, and body mass, which is a condition-dependent variable. First, model comparisons support that POT1 gene expression was a much better predictor of first egg date than telomere length, where earlier breeders exhibited lower POT1. Likewise, female body mass was also better predicted by POT1 gene expression relative to telomere length, with heavier females exhibiting lower POT. Taken together, we can see that females with lower POT1 gene expression bred earlier in the season and were larger in body size, two traits associated with higher reproductive output. For the next question, I took an experimental approach to ask whether telomere length or POT1 gene expression better predicted resilience to stress. I injected chick-rearing females with lipopolysaccharide, or LPS, which induces a temporary immune response that typically leads to lethargy and decreases in parental care. I then tracked changes in provisioning rates to their nests on the day before and during the stressor. Blood was used for telomere length and POT1 metrics and were sampled immediately before injections. Not surprisingly, the stressor led to significant decreases in provisioning rates. When we look at the relative predictive power of telomeres versus POT1 gene expression on changes in these provisioning rates, POT1 vastly outperformed telomere length. Here we find strong support for a treatment by POT1 interaction, where within the LPS injected females, those with the lowest POT1 gene expression were best able to maintain control-like visitation or provisioning rates. So consistent with our findings on female reproductive quality, lower levels of POT1 are again associated with higher performance. In this case, this putatively more protective phenotype of lower POT1 is associated with resilience to stress and may help buffer the body against wear and tear over a lifetime. Now, when I injected moms with LPS, I also induced an intergenerational stressor on their chicks that temporarily slowed their growth. This manipulation occurred when chicks were 5 days old, at a time when they're growing, 25% of their adult weight within just a 2 to 3 day span, essentially almost doubling their mass during this peak of growth. Previous work has connected early life growth to telomere dynamics, and using this scenario, I can ask whether telomere length and POT1 are responsive to the stressor and assess any downstream fitness consequences. From each chick, I sampled blood immediately following maternal injections, and again about a week later when chicks were 12 days old. I then quantified POT1 at the end of the study and changes in telomere length over the study period. I found that these chicks of LPS injected moms had significantly lower POT1 gene expression at the end of the study, and interestingly also showed parallel buffering or even elongation of their telomeres. Putting these two results together may suggest that during stress, decreases in POT1 could allow greater telomerase activity and therefore poised telomeres for repair and elongation. So I next asked whether these stress responsive traits predicted 2 fitness milestones in chicks, including successful fledging from the nest and recruitment into the breeding population. Model comparisons show that telomere dynamics and POT1 gene expression were not predictive of fledging success. Not shown here, variation in fledging was instead linked to other variables like body size and stress reactivity. However, we have some evidence that telomere length and POT1 gene expression predicted recruitment into the breeding population, where chicks exhibiting lower POT1 and telomere elongation were more likely to be recaptured in a future breeding season. So again, we're seeing this higher performance traits that are perhaps connected to a poised for repair phenotype, namely low POT1 and telomere elongation or buffering. So to summarize, I started to explore whether variation in the shelterin protein POT1 maps onto fitness-related metrics in a wild system. I found that POT1 gene expression was often more predictive of these metrics than telomere length itself. And I found that lower POT1 consistently predicted better performance, including higher reproductive quality, resilience to stress, and survival. This may support that telomere length gains its biomarker status through the pleiotropic roles of its regulatory mechanisms. In this case, shelterin proteins may in part control both telomere length and body condition, thereby creating these indirect correlations between telomere dynamics and fitness or senescence. At this point, we know very little about shelterin proteins in wild systems. So in order to explore whether natural selection can act on these mechanisms and generate the incredible diversity of senescence we see in nature, we need to place working knowledge of shelterin proteins from biomedicine into an ecological context. So where I think the field needs to go next is almost start from scratch to first quantify standing variation in shelterin proteins across groups and environmental gradients. Similarly, the degree of shelterin protein heritability, as well as their plasticity following a variety of natural stressors, will shed light on the mechanistic underpinnings of senescence. Direct and indirect manipulations of shelterin proteins via generalized stress regimes or genetic modification will bring us one step closer to questions of causality. And, aging is clearly not a single gene process, so shifting the emphasis from single mechanisms to entire pathways, using methods like RNA-seq, for example, may provide insights into both stronger and weaker integrative effects of multiple biomarkers, many of which we've heard about today. And this can allow us to truly assess the building blocks contributing to the evolution of senescence. The telomeres quickly become a face of senescence, but we need an increasing emphasis on the regulation of telomeres, because only then will we be able to peek into this black box at mechanisms that truly drive the evolution of senescence. So with that, I'd like to thank all of the amazing support from the Roswell Lab, as well as my funding sources for making this project happen. I am currently finishing up my PhD and am actively looking for postdoc opportunities. So I'd love to continue the conversation about how to fit shelterin proteins into the framework of senescence. So please feel free to contact me for a chat. Thank you all for coming, and I'm happy to take any questions. Thanks.